Welcome to the Neural Implant Podcast, where we talk with the people behind the current events and breakthroughs in brain implants in an understandable way, helping bring together various fields involved in neuroprosthetics. Here is your host, Ladan.
Hello, everyone, and welcome to the Neural Implant Podcast. Today, we are going to be doing another, I guess you could call it like a COVID series thing where we summarize a summit or a conference that happened. And this one was put on by Feinstein Institute for Medical Research, which is in New York. We've had a lot of guests on from there. I think this will be basically the fifth one that we've had on the show. But they recently had their fourth annual Bioelectronic Medicine Summit, and that was end of September. And basically, we're just going to be talking to Dr. Sangeeta Chavan, and we're going to be talking about a summary of this and the key takeaways of this conference. And again, this is a quick summary overview. If you're listening, you were one of the speakers, or there was especially a speaker that you want to hear about, we don't want to make this really long. This is essentially like a 9 or 10 hour conference. So we want to do a quick summary of this. So don't be offended, please. It's nothing personal. But I really enjoyed this conference. I really enjoyed this summit. As always, it's better to attend these in person. This would be a really good one. Hopefully the one in 2 years in Sweden will be done in person. But anyways, hopefully you enjoy. See ya. Sangeeta Chavan, pleasure to have you on the show. You are, I guess, the representative who's going to be talking about the Feinstein Institute for Medical Research Bioelectronic Medicine Summit, which actually took place in late September 2020, and we are going to be doing a summary of this 2-day summit. There were some really big speakers, Dr. Tracey, of course, who helped found the Feinstein Institute, as well as, maybe a dozen or two other really big speakers. And it was a really good conference. I'm very honored to have attended. But for those that weren't able to attend, because it was a paid conference, we wanted to give a little bit of a review of what happened. So first of all, Sangeeta, thank you so much for coming on. And do you want to introduce yourself and the conference a little bit better than I just did?
Sure. Thank you, Ladan. Thank you for having me over here. I'm a professor at the Feinstein Institute for Medical Research, which is the research enterprise for Northwell Health. We are at Manhasset, New York on Long Island. And we are talking about the Bioelectronic Medicine Summit that we did on September 23rd and 24th. So this was supposed to be an in-person conference originally, but due to the current situation, we switched to the virtual conference. And I think it was a good decision to switch to that. And the conference was great, as we'll discuss shortly.
Excellent. Yeah, it was unfortunate that it wasn't able to be held in person. I was looking forward to it. I would have loved to have attended.
This was the fourth conference, actually. We had 3 other conferences in person. So we have this one conference in New York, and then we have one in Stockholm. So we have been doing that for past few years now. And the next conference has already been announced, which will be in Sweden again. So you're welcome to join us.
Excellent. Yeah, but they're not every year. They're every other year,
they're
right?
Every other year, yeah. Every other year, one in New York, one in Stockholm.
Excellent. I will be sure to attend. Okay, so let's go a little bit into this. I guess let's start with the big headliners, Dr. Tracey, and what he talked about, and then maybe what we talked about a little bit, or what the speakers talked about on the first day.
Sure. So Dr. Tracey, he introduced the conference. He started with a meeting. Dr. Tracey has pioneered the field of bioelectronic medicine, as most of us know, basically. So he has a strong research background into neuromodulation, and specifically neuromodulation of inflammatory responses. His preclinical studies have been published extensively in very high impact journals, and that has paved the way for clinical research. And those clinical trials are ongoing. Some of them have completed, and those have been successful trials in rheumatoid arthritis patients and Crohn disease patients. So we did have 2 speakers who are running those trials, so I will touch upon that. The way the meeting was organized was we tried to cover different areas into bioelectronic medicine. So right from materials to clinic. So what is bioelectronic medicine? So bioelectronic medicine is a relatively new field where we are trying to target nerves and trying to stimulate or modulate the neural activity for the benefit of diseases instead of using drugs. So as we all know, drugs have side effects, right? So here what we are trying to do is we are trying to target nerves in a patient-specific manner, disease-specific manner, so we can reduce the side effects. To do that, then there is a lot of different steps involved in that. First, we have to identify a target. We have to identify a molecular mechanism that is leading to that particular disease. Then we have to identify the neural circuit that controls or can regulate that mechanism, and then devise a therapy or device that can modulate that nerve activity. When you take it this whole into ensemble, that is called a bioelectronic medicine. So bioelectronic medicine has three different arms. One is the engineering, one is the medicine, or one is the molecular target. To cover all of these aspects of the bioelectronic medicine, we had different sessions in this meeting. So the sessions covered about different materials that can be used to make these devices. So that was a material science session. Then we had speakers who discussed different neural circuits which are underlying these diseases. So specifically in that session, the speakers discussed neural circuits in cardiovascular diseases. We had actually two sessions, one on each day, where the speakers discussed the advances in bringing bioelectronic medicine to clinic. So they discussed about different diseases, Crohn's disease, neuroplasticity, and Dr. Yosher from Texas, he discussed about for vision. So there were different clinical studies, those were discussed in those clinical trial sessions. Then we had a very interesting talk from our keynote speaker, Dr. Larry Steinman, and he talked about different amyloid proteins which are playing a guardian role in different inflammatory disease models. And he discussed the journey about identifying this protein and then studying the role of this protein into different inflammatory disease models.
Okay, yeah, very cool. I think this breakdown was good. I do have to say, because I am a little bit more personally in the fabrication side of things, I really like the first session, Material Science and Electronics, which was chaired by Chad Boughton and Lauren Reith, both of which were previous guests on this show. And yeah, just getting into like how to fabricate these neural devices. And so I thought that was very interesting.
I didn't mention that, I guess. There was another session or another speakers where they talked about the non-invasive neuromodulation. So when I'm talking about rheumatoid arthritis or Crohn disease, there we are using invasive vagus nerve stimulation or vagus nerve stimulators, right? Implanted onto the cervical vagus nerve. But then there was Chris Puleo from General Electric, where he talked about non-invasive neuromodulation using ultrasound as a therapy. And his team has done extensive studies using this ultrasound into both inflammatory disease models as well as metabolic disease models. And they have published a few papers in that particular space. And when you target the liver site, or the neural plexus at the liver, you can really modulate levels into the subject. So they have done mainly animal studies where they have used different diabetic disease models, either diet induced or genetic diseases. When you target the liver, you can really control or you can really regulate these circulating blood glucose levels. And then they have carried out extensive studies to map where these signals are going to when you stimulate on the liver, and they have mapped the circuits in the brain which have been regulated by the glucose uptake or glucose increase or decrease levels. So that was a very interesting study too. The first day, another very interesting talk was by Dr. Reneker, who is from Texas again. He talked about targeted neuroplasticity. So that was an interesting approach too. They have brought these preclinical studies to clinic in very short time, that less than 7 years, I would say, where they are doing vagus nerve stimulation. Now, in this case, invasive vagus nerve stimulation, and they are giving rehab training to these different diseases. And diseases can range from spinal cord injury or ischemic stroke or any traumatic brain injury, etc. And they show that when you pair vagus nerve stimulation with a rehab, the animals learn faster and they recover faster. And they have brought this to the clinic now, and they are doing these clinical studies where they are pairing vagus nerve stimulation with rehab training to see whether the plasticity can be improved for those patients.
Yeah, it's very fascinating stuff. Actually, I talked to him when I visited in Texas, the University of Texas' Dallas. A lot of stuff happened on the first day, a lot of fascinating topics, and definitely learned a lot. I guess, what was your favorite topic or maybe speaker or session for the first day?
My favorite was, I would say, it was combining speakers between different sessions. Like I really liked Molly Stevens' talk too. where she talked about different material science approaches, because she's targeting different aspect of it, not just making devices, but looking at how the devices are talking to the tissue. So that was very interesting. I know Chris Puleo's work, so that talk was very fascinating also. And I think another very interesting aspect that was touched upon on the first day was by Stavros, and then it was also touched upon on the second day by by Kip Ludwig too. That when we are doing all these animal studies, as we call the preclinical models, we always have to remember how we can translate what we learn in animals to humans. And that was really brought upon by these guys was to show that vagus nerve is so different between mice, and that particular difference was very interesting to know and learn. Their study showed that mice and rats, which are the most common animal model used in all these preclinical experiments, they have a different vagus nerve morphology or anatomy as compared to human, very different rather. But interestingly, swine models or the pig models, they are very much closer to human. So maybe it is really important to think when we are developing this technology, we should always develop it from the small animal models to the large animal model, and why it is important. So that was a very interesting topic that was touched upon on day one. I think I already talked about Dr. Eneka's work, where he touched upon bringing this again preclinical studies to clinical observations and the clinical trials they're doing. And then the last one was a clinical trial update using vagus nerve stimulators into RA patients and then Crohn's disease patients. So it was interesting across the board, I would say.
Yeah, fascinating stuff. And as they say with the rat studies, is like cancer's been cured thousands of times in rats, but the important part is to bring it over to humans. So I think this is very similar to that. Excellent. So, Dr. Chavan, let's move on to the second day. What was kind of the overview of that? How did it start off? What was the keynote speakers? Who were the big names on the second day?
So the keynote speaker was Dr. Steinman, as I said. And Dr. Steinman talked about amyloid proteins and the role of amyloid proteins in different inflammatory disease models. And that was very interesting also, the way he gave the overview of past more than 2 decades of work, and where he showed that how they have identified these amyloid proteins. And they call it as, or rather he calls it as guardian molecules, because they are actually playing a key role in preventing inflammation. So if you knock out these proteins into again animal models, there is exacerbated inflammation. And if you come up with these proteins, you can again cure different types of inflammatory insults in these models. And again, he has used different animal models, and he and collaborators have used different animal models like ischemic models, optic neuropathy, to show that these amyloid proteins, and again here I'm talking about amyloid precursor protein, to show how they prevent inflammation in these different animal models. And they have identified the motif that is present in this protein that is responsible for this activity, that is a hexapeptide. And then they have gone ahead and shown how the hexapeptide is acting and what is the molecular mechanism underlying these anti-inflammatory effects of this particular protein family. And they showed that these protein families are binding to different inflammatory molecules, and that's how they are preventing or they are regulating or suppressing the inflammation. So that was a very interesting talk during this meeting, and that was our keynote talk also for the talk for this particular meeting. And then that was followed by different developments in bioelectronics field. As I said, Dr. Kip Ludwig, he talked about again vagus nerve stimulation and different advances which are happening in that particular field. Dr. Larry Miller talked about how you can modify. I'm blanking on the word.
I'm looking at it. It's sphincters. I remember that one actually. It was really funny because he's like, I'm the only sphincterologist in the world.
Correct. So he is using the sphincter, he is using neuromodulation technique to modify the sphincter opening and closing, and which is really important in different diseases. Though he is looking at that particular technology can be utilized to different valves, and that's what he has been studying extensively on. Then that day, so we had 2 sessions and one keynote speaker session. So the second session or the last session in that day was again another clinical trial update, where Dr. Weber gave us an update on his haptics program. And then Dr. Yosher gave us an update about prosthetics for vision. So that was an interesting session too.
Yeah, very cool. So yeah, it was a very interesting conference and a lot of information there. I very much enjoyed it. There's a pre-recorded lectures and then there was a Q&A. And so you were able to ask questions and everything like that. Yeah, what are the takeaways that the listeners should take away? What do you think that they should remember about this Bioelectronic Medicine Summit?
Okay, what the viewer should remember is that we can target nerves to cure diseases. That has been studied for many years now. And those studies in rats and mice, as we said earlier, are being brought to patients. And we are seeing positive effects or positive results in different patients in different disease scenarios, not just one disease scenario. So definitely the bioelectronic medicine field, relatively new field, it is growing and it is really succeeding. So I think that would be the take-home message over here.
Excellent. Yeah, it's really inspiring. I really like it. Okay, very cool. Well, thank you for this summary of the conference. Does Feinstein have anything coming down the pipeline in the next months or years?
Yeah, Feinstein Institute is one of the pioneers into bioelectronic medicine field. Basically, based on our studies, based on Dr. Tracey's studies, the field has started. And we have an Institute of Bioelectronic Medicine here where we have many leaders who are the faculty over here. And so we have a very collaborative atmosphere across these different faculties, and we have different expertise. Like we have engineers on board, we have biologists on board, cardiologist on board, or gastroenterologist like Dr. Larry Miller, who talked about the sphincters in this meeting. Then we have basic scientists on board. So we are doing both preclinical and clinical studies at the Feinstein Institute. So if I talk about what is in the pipeline, is there are actually many things which are ongoing in the pipeline, because there is deep brain stimulation happening in the clinic, which Dr. Ashish Mehta is doing, or other lot of preclinical studies which are happening both in the rat or mice model as well as in swine model, which will be brought to the clinic very soon. So definitely there is a lot to look upon. Then there is Chad Boughton, who is working on paraplegic patients. So there is definitely a lot of different aspects which are happening in the Feinstein Institute.
Very cool. Very cool. It's not a secret that I like your guys' institute. I think you are the fourth or fifth guest I've had from there on the show. I very much enjoy what you guys are up to. Dr. Chavan, is there anything that we didn't talk about that you wanted to mention?
No, I think we covered the meeting. Thank you for having me, Ladan. Yeah.
Hope you enjoyed the show and were able to learn something new, bringing together different fields in novel ways. Until next time on the Neural Implant Podcast.